In this video, we are going to start talking about gene regulation in prokaryotes. And when we talk about gene regulation in prokaryotes, there are two key models to talk about, and that is gene regulation of the Lac Operon and gene regulation of the trip operon. And we will cover the Lac Operon in this video, and we will save the trip operon for the beginning of next lecture. So the Lac Operon, what the heck is the Lac Operon? We haven't even talked about operons yet. I try to do mostly eukaryotes in this course. I work with eukaryotes, and it's kind of hard to cover both. But let me first talk about what lactose is. Okay, when we talk about the Lac Operon, we're going to be talking about a set of genes that controls lactose metabolism in prokaryotes, definitely E. coli. So lactose is a disaccharide. Essentially, it's a sugar molecule, and it's composed of galactose and glucose, and these are linked together with some fancy bond there. And there's an enzyme encoded by E. coli called beta-galactosidase. What beta-galactosidase does is it breaks this bond between these two and frees up galactose and glucose. So now we have free galactose and free glucose. In E. coli, in most cells, they really want glucose. They want to get at the glucose because that's a really good source of energy. So the Lac Operon is going to be involved with genes that are required to utilize lactose to get at that glucose. And you're not always going to want the Lac Operon to be expressed. Like when glucose levels are high, we don't want the Lac Operon to be expressed. Or if lactose levels are low, you don't want, or E. coli doesn't want that Lac Operon to be expressed. And so what is an operon? Well, in prokaryotes, genes are organized, many genes are organized into these things called operons. And so what is an operon? It's just the situation where you have multiple genes under the control of one promoter. So this is something new. We haven't seen that yet. We were talking about transcription. You know, we talked about promoters. We always put one promoter in front of one sort of coding sequence. Now, in prokaryotes, many of the genes are organized into operons. And the one we're going to cover now is called the Lac Operon. Now, are there operons in eukaryotes? I'm sure there are some somewhere, but they're not very common. Not very common, as far as I know. And I need actually to look more into that. The most important thing to know is that operons are common in prokaryotes, not common in eukaryotes. So, the Lac Operon, what does it look like? Well, there are three key, what we're going to call structural genes. And what is a structural gene? So a structural gene is any gene that's not a regulatory gene. Well, what is a regulatory gene? Well, let me start with a diagram of the Lac Operon first. So we have LAC-Z, LAC-Y, and LAC-A. Now these are three genes, structural genes, and LAC-Z encodes beta-galactosidase, so that enzyme that catabolizes lactose into galactose and glucose. This is a permease, and this protein right here is thought to increase, how lactose get into the cell, or increase the efficiency with which lactose gets into the cell. And LAC-A encodes a trans acetylase. So we don't even know what Lac A does. Even though it's part of the Lac Operon, scientists aren't really sure what role this has with respect to lactose utilization by the cell. So these are all structural genes. And that's just a way to classify a gene. So a gene that is not a regulatory gene is called a structural gene. So a regulatory gene is any gene that makes a protein that is involved in gene regulation, regulating other genes. So determining whether those other genes are transcribed. So beta-galactosidase, that's an enzyme that doesn't really influence whether another gene is transcribed. Permease, no, helping lactose get into the cell, doesn't really or directly regulate another gene, and we don't even know what this does right here. So I guess that could be a regulatory gene, but for the most part, we are saying all of these three right here are structural genes. Now, that's only part of the Lac Operon. So let's say the Lac Operon ends right here. Now there are two other regions. Now these aren't really genes, but we give them gene-like names so we can talk about them. Lac P, we're going to call that the promoter. It's not really a gene, and we call it Lac P. It's I should probably just call it P, but we'll call it Lac P for the promoter region, and Lac O for the operator region. Now, we haven't talked about operators yet, so these are as far as I know, specific to operons. So, operator. Now, this is the promoter right here. Okay, so here we have the complete Lac Operon right here. Now, there's another important component that is involved with regulating the operon. It's not part of the operon, but it's involved with regulating the operon, and that's a regulatory gene, and it's found somewhere else in the E. coli genome. And I'll indicate somewhere else by putting this little, whatever you call these little diagonals here to indicate that it might not be necessarily right next to the operon, somewhere else in the genome. In this gene, I'm not even going to diagram its promoter or anything. Let's just know that it is always expressed. So let's say this thing must always be expressed in the cell in order for this operon to be regulated properly. So Lac I, what does it encode? It encodes the repressor, the Lac repressor. It should be called Lac R. That'd be better, right? I don't know where this name came from. Maybe Lac inhibitor. But it's the Lac Operon repressor, just for short, Lac repressor, Lac repressor. Okay, so it's a repressor protein. And let me see. So, and we can diagram the repressor protein to look something like this. Okay, so there's the Lac I repressor protein. And we have these two binding sites right here. This right here, this is going to be the lactose binding site. And we can diagram lactose like this. Okay, so this is our lactose molecule. It doesn't look anything like that, but this represents lactose. And this is the lactose binding site. This right here, this is going to be the operator binding site. I should specify Lac operator binding site, or just Lac O binding site. This is the part of the Lac I repressor, the Lac I protein, that's going to bind the Lac O operator, the binding site. So it's a specific DNA sequence in the operator region of the Lac Operon. So more about this promoter right here. So let's say this is where the +1 site is. I don't know for sure if this is like, if the +1 site is somewhere in the operator or it's in the promoter. But let's just assume it's right here. So RNA polymerase, RNA-P would bind here, transcribe this. And when it transcribes that, what are we going to get? We're going to get what's called a polycistronic messenger RNA. Polycistronic mRNA. So what is a cistron? A cistron is a really old term, or maybe it's common for people who work with prokaryotes. I think it's a term that's kind of going out of use, but maybe not. Maybe microbiologists would say, no, we still use this. But anyway, cistron means section of DNA or RNA that codes for a protein. Okay, so polycistronic means there are multiple cistrons. mRNA, that's kind of redundant, right? Because if a cistron codes for a protein and a messenger RNA is an RNA that codes for a protein, we probably don't need the M. So polycistronic RNA is a messenger RNA with multiple cistrons. So with this messenger RNA here, it might look something like this after transcription. So we'd have the Lac Z region with a start codon and a stop codon. We'd have the Lac Y region with a start and a stop, and the Lac A region, again, with a start and a stop. So ribosomes will load on the 5' untranslated region, or right over the start codon. We didn't really focus too much on those details in this course. Start translating, and then it will get to the stop codon, and then we'd have that Lac Z protein produced, and we can diagram the Lac Z protein something like this. Now, another thing we don't really talk too much about, mostly because I don't really know, I forget these details, but does the ribosome stop here and then keep going and look for the next start, or does another ribosome just assemble right over this start right here? I don't really know, and I don't expect you to know. I won't test you on that. You might cover that in microbiology or cell biology. But the important point is that ribosomes will translate this, and then either whether they keep reading, and restart again, or maybe this is an internal ribosome entry site and ribosomes load on here. This Lac Y will be made, and I can diagram the Lac Y protein to look something like this. This is the permease. Maybe the lactose goes right through there. And same thing for this one. And Lac A, we don't really know what that does. Transacetylase, but we don't really know what it's doing with respect to lactose metabolism. So we'll give it a shape that looks like this. So, okay, so we learned a little bit about polycistronic RNAs. We learned what a cistron is. We learned that the Lac Operon is, or what an operon is in general, right? So an operon, multiple genes under the control of one promoter. RNA polymerase loads, transcribes all of this, all these cistrons on one RNA. All the proteins can get translated from that one RNA molecule. Okay, so we learned a lot of things here, and now we have to get back to, how is the Lac Operon regulated when lactose is present and when it's not present? So this is going to be the most important right here. We will talk a little bit about how glucose is going to play a role later, but for this part, when we're just talking about lactose levels, the Lac repressor is the most important. So now, the Lac repressor, called Lac I, I wish it was called Lac R, but it's called Lac I. Now, it is an allosteric protein. And these proteins, allosteric proteins, are common when they're involved in regulation. Allosteric. So that means its shape changes depending on molecule, whether a molecule is bound to it or not, a molecule that is supposed to bind it. So allosteric protein. So when lactose is bound, remember this is our shape that symbolizes lactose, and it's not to scale, right? Lactose is a relatively small sugar molecule compared to this protein, so not to scale. But that operator binding site goes away because the protein structure shifts undergoes an allosteric shift in that binding site. That site it needs to bind the operator disappears. So it can no longer bind the lac operator. Now that's when lactose is around. But if lactose is not around, well, it looks like this. And you can see when lactose is not around, it can't be bound to the lactose binding site. And so the protein adopts a structure that opens up that lac operator binding region right here. Allosteric shift. Every time I diagram these things right here, this reminds me of a torso, of like a person, right? And my daughter and her friends had a sleepover the other day where they made a movie called Just the Torso. And the reason they did it was my wife makes skating dresses for my kids who are ice skaters. And she has this little thing to hang the dresses on, and essentially it's just a torso. And it's pretty creepy looking thing. If you're like, we store it in the basement and we walk down and you see it down there, it's like, oh, it's just a torso. So they made a movie, Just a Torso. It was pretty cute, a little scary. Anyways, what were we talking about? Oh yeah, so the allosteric shift. So when lactose is not around, the operator binding site opens up. Okay, so now we're ready to talk about how the Lac Operon is regulated. And I'm going to cheat a little bit and save some time, since I just wasted some time telling you about my daughter's Just the Torso movie. So when lactose is present in the cell, lactose is bound to lac I. The lac operator binding site of lac I is not available, not there, so the repressor cannot bind to the operator. And when it's not bound to the operator, RNA polymerase can transcribe through the operator region. Let's say a +1 is here. Transcription start site is here. It can transcribe through, and then it can transcribe all of these genes, lac Z, lac Y, lac A. And remember, transcription in E. coli can occur at the same time as translation, so those ribosomes can hop on and start making proteins. So that's when lactose is present. What about when lactose is absent? So when lactose is absent, well, lac I is bound to the operator, and that can physically block RNA polymerase should it be bound to the lac promoter. Now there's some question or debate whether or not it's actually blocking RNA polymerase, or maybe when it binds to the lac operator, it causes the DNA to adopt such a structure that RNA polymerase can't bind anymore. But long story short, right, is whether it's blocked or it's preventing it from binding, transcription does not occur. So when lactose is absent, transcription does not occur, and that's important because you don't want to be making these genes right here, at least lac Z and lac Y, which are required to catabolize lactose and lac Y to help more lactose get in. How does that initial lactose get in? Well, maybe it leaks in or whatever, but the permease will make lactose get in even more efficiently. So, okay, so this is how E. coli regulates transcription of the Lac Operon when lactose is present and lactose is absent. So there's some other things too. So I hope that made sense. And now another problem is glucose, right? So E. coli really prefers glucose over lactose. So glucose, I'm going to put exclamation points here because it wants this. And if this is at high levels and this is at high levels, E. coli apparently doesn't want to waste time with lactose because it has evolved mechanisms to shut down the Lac Operon when glucose levels are high, even if lactose is high. So how does that work? So let me just summarize that. Shut down Lac Operon when glucose high. Even if lactose is high, right? So far we just talked about whether lactose is high or lactose is low. And with the information I've given you so far, it's as if, you know, lactose is the only thing that matters with respect to regulation of the Lac Operon. But no, there are other reasons why E. coli might not want that lactose operon transcribed, even when lactose levels are high. So to understand how glucose levels play a role, what we need to do is look a little closer at that promoter region of the Lac Operon. So let's say this is the Lac P region right here. Now I can divide that into two parts. So one part is going to have what we're going to call the cap binding, is going to be the cap binding region. Cap binding, like baseball cap binding region. And then right here we'll call this, I'm not going to draw any fancy structure on it, the RNA polymerase binding region. So two parts of the promoter, and here's the operator over here. Lac OC. And in case I forgot, so these little knobs I'm putting here, so these are just DNA sequences, and we're putting these little knobs on here just to represent that there's a sequence there that a protein binds, and it's located at this region right here. Now, cap binding site, what is that? So essentially, there's going to be this protein called cap. It is an allosteric protein, and it's going to bind the cap binding region when it is bound to another protein, a molecule called cyclic AMP. And I'm going to diagram the cyclic AMP like this. And again, this is not to scale. I'm going to tell you what cyclic AMP is in a moment, and no way is it as big as the cap protein. But this is a cyclic AMP, little c, AMP plus cap complex. And together, they bind the cap binding region. And when this is bound to the promoter, then RNA polymerase can be loaded onto the promoter. I guess it's not loaded very efficiently on the promoter when this is not there. So if this is present, RNA polymerase can be put on the promoter, and then it can transcribe the Lac Operon, assuming the Lac I protein is not here. And when would the Lac I protein not be bound here? When lactose was bound to it. Okay, so pencil's getting a little dull. So we need to understand how glucose influences how the Lac Operon is transcribed. We need to understand how glucose controls whether or not this is bound here. So this is relatively easy. So glucose levels are low. It's easy because we're not going to talk too much about the details of how this works, because I imagine the details are really complicated. But somehow, when glucose levels are low, what am I diagramming here? I have no clue what this looks like. But what I want it to be is an enzyme called adenyl cyclase. So adenyl cyclase is an enzyme that takes ATP, so adenosine triphosphate, or adenine triphosphate. So just the ATPs, a ribonucleic acid right here, ATP, the same thing needed to make, this is a ribonucleic acid, not a deoxyribonucleic acid, so the same thing needed to make RNA, right, to transcribe RNA. This is one of those. This is an ATP with 3 phosphates. And adenyl cyclase takes that and turns it into cyclic AMP. So a molecule called cyclic AMP. Now, when glucose levels are low, the adenyl cyclase activities are high. So glucose levels low, adenyl cyclase activities are high. So like I said, I'm sure the details are complicated. So how low glucose levels increase the activity of this, we're not going to talk about. I imagine it could be pretty complicated. So ATP is converted to cyclic AMP at high levels. This rate is high, so we get high levels of cyclic AMP when glucose levels are low. Now I'm going to diagram cyclic AMP like this to go with our previous diagram. And then we're going to put that together with our CAP binding protein. CAP stands for catabolite activating protein. Now, when these are high, there's a good chance it's going to find CAP here, and they will form a complex. And like I said, CAP is an allosteric protein, and it undergoes an allosteric shift. And I should have put it hatched marks to keep this consistent with this guy over here, but I didn't. But it's going to undergo an allosteric shift, and it's going to open up that Lac promoter binding site, the region of the protein that can bind the Lac promoter here. Okay, so it's ready to bind to the Lac promoter. Now we have the opposite situation, right? Now this could be kind of complicated because I think cells need a little cyclic AMP no matter what to survive. So we can't totally shut down this process. But what we will say is that when glucose levels are high, adenyl cyclase levels are low, adenyl cyclase levels low. And so low levels of cyclic AMP. It's easiest just to pretend those don't even exist anymore. But like I said, I think cells need some cyclic AMP for survival. I'm not sure on that, but easiest to think that there's not much of this around at all. And when there's not much around, the CAP protein, which we consume is always around, is always transcribed, doesn't bind to cyclic AMP, doesn't undergo the allosteric shift, and it's unable to bind to the promoter region of the Lac Operon. So again, I'm going to cheat to wrap this all up. So when glucose levels are low, and lactose levels are high, what's going to happen? Glucose levels are low, cyclic AMP levels will be high. They can form a complex with a CAP binding protein that can bind to the Lac promoter. When that's bound to the Lac promoter, it helps RNA polymerase bind to the promoter. And so this can be transcribed. The operon can be transcribed as long as lactose levels are also high, right? Lactose levels are high. The Lac I repressor protein, the Lac I protein, cannot bind the Lac operator. So what about glucose high, lactose high? If glucose is high, well, the cell doesn't want to express the Lac operon because it's got plenty of glucose to use. It wants to use that first. So cyclic AMP levels are low. CAP is not bound to cyclic AMP. It cannot bind the promoter, and RNA polymerase does not bind the promoter either. So the Lac operon is not transcribed, even though the Lac repressor is not blocking transcription of the operon. Why isn't it blocking it? Because lactose is high.